All right. Our next presenter is Allison Lee. Allison Lee came to the MAS program after working 10 years in research, the past 7 of which involve studying phytoplankton's genetic response to climate change and ocean acidification in the lab and at sea. A moment that sums her up well. She ran a marathon in Antarctica, dressed as a banana, the same day she boarded an icebreaker for a 2-month expedition at sea. Pretty interesting lady over here. Please join me in welcoming Allison Lee, who's going to talk to us about phytoplankton, citizen science, and Antarctica.
Hello, thank you for being here. Talk amongst yourselves while I get this going. Okay, I think I'm actually going to be a pioneer and be the first today to use the laser pointer. So, and before I start telling you about using citizen science in Antarctica, I think it's important that I first talk to you about phytoplankton, Antarctic 101 style. So, journey with me to the most remote region in the world, the Antarctic continent, where winds and currents swirl around, making it the windiest, the coldest, and the driest region in the world. The coast on the peninsula is lined with fjords, and fjords are just deep-sided valleys that were cut by glaciers. These fjords are hotspots for biodiversity, and they provide habitat and refuge for whales, seals, and penguins. And these charismatic megafauna are supported by what I like to call the charismatic microfauna, or the microscopic plants and animals named phytoplankton or zooplankton that hide within the watery world. The phytoplankton in this invisible forest play a critical role in contributing to the Earth's atmosphere. As they use carbon dioxide during photosynthesis to produce oxygen, they actually produce over 50% of the Earth's oxygen. So that's more than the trees and the plants on land combined. And this video is showing carbon absorption by the plants in the ocean and on land, but it mirrors oxygen production, and it's almost as if you can see the Earth breathing as the two act like lungs. These organisms, phytoplankton, are also used as sensitive indicators of a changing environment. And one of the fastest changing warming regions in the world is the Antarctic Peninsula. Over the past couple of decades, there's been a significant amount of ice mass loss, which you can see here in red all along. And you might think, if Antarctica is so far away and barely anybody goes there, then why should we care? But this amount of ice loss is contributing an influx of fresh water into the oceans, and that's contributing to global sea level rise. But more locally on the peninsula, we don't quite know how this fresh water coming into the fjords, which are those transition zones between the land and the sea, are going to influence the biology. So as that meltwater, we call, comes in, it can change the physical and the chemical nature of the water, and that could influence this phytoplankton. So if the phytoplankton change, it could influence the things that eat them, the krill, and if the krill change, then it could influence the things that eat them, the whales, seals, and penguins. So if you disrupt one of these groups, you could influence the others. Scientifically, we're interested in understanding how the presence of this meltwater exists along the peninsula from the north to the south, as well as from those fjords into the offshore environment. We're also interested in understanding the timing of the phytoplankton blooms throughout the full season. So at the bottom of the world, the summer is November through March. It's opposite of what we experience here. And we're also interested in understanding the composition of the phytoplankton blooms. So just like wildflowers blooming in a field, not all fields have the same flowers. It's similar, it could be similar with the fjords. Not all of them have the same composition of phytoplankton. And fjords in Antarctica are understudied systems, and this is complicated by the fact that it's a very remote, harsh environment to operate in. It's also logistically very expensive. So we should be creative in how we leverage the existing networks that are down there. And you may be thinking, well, who's already down there? Antarctica's only recently been explored since 1820. And in 1959, it was set aside by multiple nations as a place entirely devoted for peace and science. So in the summers, you have around 4,000 science and support staff operating down there from 29 different countries, as well as a fishing industry. But you also have a very large and active tourism community. And tourism is self-governed under the International Association of Antarctic Tour Operators, or IATO. And commercial tourism started around the peninsula 65 years ago, give or take, in the late 50s. And it's mainly focused on the peninsula. So in red, you can see the vessel traffic density. And in the early 90s, you had maybe 6,400 visitors to the peninsula. And just this last season, there were 44,000 visitors to the region. And partially this is due to increasing economies and closely matches a country's gross domestic product, but it's also indicative of that ice melt and that these previously once closed waterways are now open as the ice has disappeared, and you don't need big icebreakers to go through these areas. So the tourism industry has a presence there through that austral summer, the November through March. And the popular tourism sites are actually within these fjords scattered all along the peninsula. It gets lighter red down here, you can see. So it could be a unique opportunity to partner with this community. And when scientists join with non-scientists to do science, we call that citizen science. And a couple of the tour operators already have citizen science efforts going down. Some of the existing citizen science projects that you may be familiar with are satellites over seals and penguin watch, where you can identify individuals through online photos. There's also Happy Whale, where you can upload an image that you've taken or track individually identified whales as they move across the world. And there is also a group collecting seawater to look for the presence of microplastics. And companies are showing an interest in partnering with researchers, not just bringing tourists to walk around through the research facilities. And one of the missions of IATO is to support science. So for my capstone project the last couple of months, I've developed the Fjord Phyto Citizen Science Project, which focuses on engaging the tourism industry in hands-on sampling as they can help us understand how phytoplankton are changing throughout the season. And this, we'd provide a kit of science equipment and then train the specialist staff on board, and many of them have backgrounds in science. And then we'd be using protocols or methods that we would use as researchers in the field, but just adapted for this project so guests could also engage in the process. And this would be done through conversation, in-person training, a user's guide that I've created that they can take out into the field to reference these methods, and then also through online training videos. And the focus would be on the smaller ships that are able to access the fjords and then make those landings to shore. So as the groups journey from the main ship to the shore in a Zodiac, or a small inflatable rubber boat, they can use the simple units to take those measurements. So for instance, you can measure salinity and temperature and depth, among other things, and then deploy a plankton net, which would help concentrate that phytoplankton into a nice rich sample that you see here. And this would help us start to answer the questions of the presence of meltwater and the timing of the phytoplankton blooms as the tourists are visiting throughout that season. And then the sample would be brought back to the main ship, where it could be filtered through this small, simple setup. And on that filter, that sample, and among others, would be preserved and then sent back to San Diego, where we could do a deeper analysis at the genetic level, and as well as look under high-powered microscopes. And I've created a training video, which I plan to create others that would demonstrate how these activities would be done, because if they're going to be helping us in this project, then it's worthwhile that it's done so there could be high-quality data from these samples. And then after filtering, they would, a portion of what they caught from the plankton net, they could use to look under the microscope on board. And I think this is a really important activity because nobody is emotionally connected to phytoplankton. They are not cute and cuddly. They're alien. You need a microscope to see them. But I think once you start to realize their role in the environment and you can see them up close, that's when that connection is really made, that they're so diverse. And it's mind-boggling. I didn't know they're 50% of the Earth's oxygen or more. That's The intention with this activity is that they can take photos with their smartphone or camera, and then those geotagged photos could be uploaded to an already existing database called iNaturalist. And iNaturalist is a tool for recording observations around the world. It can be anyone, anywhere can upload a photo, and as that photo gets identified by a vetted online community, once 2/3 of that community agrees on the taxon or species level, it's moved to a research grade identification. And this part would allow that instant gratification for the people who have been involved, because it takes us a while to analyze those samples back in the lab. So for this portion, they can see that their observations are helping populate this peninsula, which currently only has one observation, and that's of a kelp, so a macroalgae. And then we can validate what they're seeing with what we see. Last season, we had our current partners, Polar Latitudes and G-Adventures, collect phytoplankton samples for us just for microscopy at the moment, and they're eager to do science. So even not all voyages are going to have the same amount of participants. Some you may only have 5 people engaged, and others you may have 15. But if one tour operator is running 12 voyages throughout the season, that's upwards of 180 people that you have participating in your science. And even if they go home and tell that story, that's really encouraging. So for the future, we are hoping that we can add the full Fjord Phyto activities to their program, and we're also interested in partnering with other vessel operators. And this project is hosted on a website that I've created, fjordphytowordpress.com, and where partners and guests can access that information and learn about the project. And then as we get the results from the lab back in San Diego, they can be uploaded for the science portion, so we can share. The project lives on even after they've taken their 10-day trip or longer. And then as part of the kit, they can download the materials checklist, the user's guide that I mentioned that they can bring into the field, data sheets, a phytoplankton identification guide that we're creating to just give very simplistic ID for the guests involved so they can understand the diversity, which could lead to educational talking points about the region, and then also those training videos that I mentioned. And citizen science is not a novel thing. There are many success stories over the past decades. But citizen science has to have a scientifically relevant contribution. And in our case, they're helping us understand how these phytoplankton change throughout the full season when we can't necessarily be there all the time as researchers. And it also has to come with engaging your participants and learning. And so as the program goes on, we can also evaluate the learning outcomes. Are they having a different perspective on science or on the environment? And I think this could be a very powerful tool to study this part of the world and also share those discoveries with the people who visit. And this has great impacts for multiple stakeholders of the region. For science, I hope I've demonstrated that you can expand the scope and supplement the data and begin to answer those critical climate questions. For tourism, they're gaining a greater understanding of the scientific process and maybe seeing the environment at a different level. They're also contributing to the research legacy that is in Antarctica. For management, resources in Antarctica are managed under the Antarctic Treaty, and it's ecosystem-based decisions. So in a rapidly changing world, the more information you have about how it will change, the better. And it also provides an example of how tourism can add value to research monitoring efforts. And then for society, you're enhancing public awareness and concern for the conservation of the Antarctic environment. So moving forward, I'm going to continue building out the website and featuring it on citizen science platforms such as citsci.org and SciStarter. SciStarter is a catalog of over 1,600 current citizen science projects that exist, and Fjord Phyto will be listed as one of them. And then iNaturalist I've talked to you about. If you want to contribute, if you have any photos from your trip to Antarctica, they can go into the Fjord Phyto project. And we'll continue writing grants and looking for funding opportunities so that we can buy the materials that we want to provide in these kits. And additionally, we'll continue the partnerships that we have and continue to build new ones. And then ultimately, these samples would contribute to what would be a portion of a PhD thesis going forward in the near future. So I'd like to acknowledge my committee members, Dr. Maria Vernay and Dr. George Waters, as well as the Antarctic contacts I had to learn more about the industry and then support I receive for attending a science conference and a citizen science conference. And then my PhD mentor, Amy Van Cise, as well as the CMBC team, the MAS cohort family, advice, mentoring, knowledge I've gained the entire year, and my friends and family. Thank you. Questions? Jayla?
So, have you looked at the samples that you got from this season?
So the samples that we got from last season are in Chile or Argentina, being analyzed under really cool, high-powered microscopes. You can see a lot of detail. So, but it takes 3 to 4 hours to do one sample. So, we haven't synthesized the results of that yet. Yes.
Hi, Allison. Great talk.
Thank you.
I'm just curious, with these kits that you're planning to hand out, what would you hope to include in them, and what would be the cost of a kit?
Yeah. So they would have a plankton net, one of those handheld CTD castaway devices that can measure salinity and temperature, as well as that little setup for the filtration. And in some cases, some of the operators have plankton nets and microscopes on board, but the idea would to have a kit that would be comprehensive of what we'd need for them to do that activity that they could do. Cost? I think I budgeted for 3 vessels to have a basic kit would be $20,000, but there's some bargain shopping to be done. So.
So I have another question from George Leonard at Ocean Conservancy, who appears to still be lurking. First, he wanted to say that he is emotionally connected to phytoplankton.
Okay. I apologize.
Second, he wanted to know how the dynamics in your system relate to those in Greenland, just published this week by Arrigo et al., where they found that melting glaciers stimulate summer phytoplankton blooms.
Well, I'll admit I did not read this week's publication as I've been preparing for this final. But I can say that Antarctic fjords are understudied in comparison to the Arctic fjords, which are well studied. And the stimulation of the phytoplankton bloom, so in the Arctic, because it's experiencing more of this cooling and warming, there's that influx of the fresh water which brings nutrients, and that nutrient content can stimulate those phytoplankton blooms. But additionally, it also causes a lot of turbidity in the water, or like mixture. So fjords up in the Arctic aren't known to be these same hot spots for biodiversity like we're finding in Antarctica. I hope that answers your question.
So you said you were looking at expanding this with research. What types of research questions do you want to dive into with this?
Yeah, so I didn't touch on this too much for this talk, but for the genetics analysis, the idea would be if phytoplankton can be used as indicators of change, there's also this genetic component where you can look at individual species more than just what you can collect in a net and look under the microscope, but additionally you can start to look at their gene activity levels and see if they're responding, as a population in any way at that level, at the genetic level, which is just a further, key for how things can be changing. Does anybody want to go? It's $20,000. Yeah, Nina?